Well, thank you for the really kind introduction, and thank you for having me here. Had a real fun day already today, learning about all the stuff that you guys are doing, and it's really amazing because this physics department has the two branches that I'm involved in, the nano bioscience, and then the cosmology branch is really heavy here too. So it's a place right after the character that of research and science that I love. All right, but today I decided to talk about, or Stuart asked me to talk about the biophysics research. And so Stuart already mentioned, I do experimental gravity with one half of my brain, and the other half does biophysics. And we were very heavily involved in getting nanopore sequencing to work. And what I'm particularly excited about today is that we developed a method that's actually better than optical tweezers to look at enzymes that move along DNA. And I will tell you a lot about this. So here's some sort of outline. I'll talk a little bit about how nanopore sequencing works, then go down a list of enzymes that we studied with this new single molecule technique. And time permitting, I will skip, or time not permitting, I will skip a few of those things. But I will definitely end up with a comparison of our technique to other single molecule techniques that are around for this type of stuff. So nanopore sequencing is in principle very, very simple. So you have a bucket that has a membrane that divides it, this membrane here. And this membrane has one hole in it. And this hole needs to be small because what you want to put through there ultimately is DNA. So you set up a voltage gradient over it with some electrodes, hook up an ammeter and measure the current. This measures the ion current, measures the current of potassium and chloride that flows through here. Then you introduce DNA, and the DNA is the A stands for acid. So for the people that are like me when I started this, acids are negatively charged. So this thing is negatively, DNA is a heavy negatively charged polymer, and it just wants to go to the plus side. So it just moves through there on its own. And while it moves through there, it modulates the ion current. And this modulation of the ion current, and what's our hope, should be proportional to the type of nucleotide that's in there. So that's a concept of nanopore sequencing. It can't get much simpler than this, but it can also not get much more naive than this either. It wasn't quite that easy. So the first thing I did in our physics department is I broke with the solid state people, and I made things out of biological stuff. And that caused a lot of confusion in the department because I was always pointed towards our nanotech facility to make these little holes. The way we do it is we make a Teflon aperture that's roughly 20 micrometers from here to there. And we span over it a lipid bilayer. So that's a component that cell membranes and so on are made of. And then we add a protein pore. And it's pretty simple because the stuff inserts itself into the membrane. When you have one pore in there, your setup is ready to go. So the pore that we concentrated on is this pore called MSPA, stands for Mycobacterium smegmatis porin A. There is a cousin of Mycobacterium, and that's Mycobacterium tuberculosis or Mycobacterium leprosy. So those are really bad disease-causing bacteria. And we saw this protein, the structure of it solved by some folks in 2004. And this seemed like the ideal pore for the job. So because Mycobacterium tuberculosis is so hard to get rid of, it was very clear that this pore is very robust. You can boil it at in the presence of some nasty detergent, and it just doesn't care. It's fine. You can put it at any pH value, and this thing is still looks like this picture I'm showing you here on the left. And DNA nicely fits through there. So the money thing about this whole pore is that it has this narrow constriction. It's just the width of DNA, and it's also not very tall. And tall is important because then the ion current really focuses just on a few nucleotides, or maybe ideally even one nucleotide. And so tougher and better than silicon nitride technology. That's only one little problem, and DNA doesn't go through it. I was about ready to give up, but then that's what I learned in physics graduate school. You don't give up on things. And so I went around and talked to colleagues, and they said, or I knew why the thing didn't work, because there was a negative charge here in the middle. And I told you just now, DNA is negatively charged. So it was just repelled from the constriction of this pore. Didn't work. And then talked to colleagues, and they said, oh yeah, it's pretty easy. You just mutate these away. So we mutated them away, made them neutral. And it was so fun that we made a whole bunch more mutations and took charges from other regions of it out also. And ta-da, this thing formed stable channels, and the mutants let the DNA through. This was then the breakthrough paper, I think, that made nanopore sequencing into something that could become reality, and was done by my student who originally was working in gravity with me, Tom Butler, and then switched to this obscure topic. So we let DNA through there, and we saw that the ion current dipped down whenever we think there was a DNA molecule going through there. Had very small dips in the order of, most of them were less than 100 microseconds long. And there was a piece of DNA going through there. It was 100 nucleotides long. And so yeah, it wasn't great, but at least we could see that this thing was a sensor for DNA. And then we ran some molecular dynamics simulation to, or yet another collaborator and colleague ran these for us. And you can see how this DNA wiggles through there, sticks here and there. And so this molecular dynamics simulation is really what is F equal M times A for simple physics. This is here is a more molecular dynamics simulation it contains the physics for biology in ways. And so this took 2 days of computer time and simulated 1 microsecond in real time. And that was really the crux. The DNA it was a confirmation that the DNA would move through there incredibly fast. So problematic, because these tiny currents that flow through the pore are in the order of tens to hundreds of picoampere. You just cannot measure a current that small without noise that totally overwhelms you, especially if you would require a bandwidth of megahertz here. Just doesn't work out. So the DNA was moving way too fast. We had to come up with a way to reduce the speed of the DNA through there. That's what we then did in collaboration with Mark Akeson. We took a polymerase. So a polymerase is one of these machines that out of single-stranded DNA makes double-stranded DNA by incorporating the building blocks of DNA and making this double-stranded DNA. So in the process, it has to pull the DNA through there. And so it does that nucleotide by nucleotide, step by step by step by step. It pulls the DNA through there. And these steps are typically in the order of milliseconds. So much, much easier to, much more conducive to measure ion currents that are happening then. So how do we set this thing up? Here comes my personal molecular dynamics simulation. So what we do is we put the enzymes first on the DNA, but the DNA has, the enzyme is parked somewhere in the middle of the DNA with some tricks that we had to apply. And there's a single-stranded piece of DNA that can, in principle, insert into the bilayer, into the pore. We did that. And here's a molecular dynamics simulation. Ta-da! This thing is on there. And so it's really, in reality, it's actually just as simple as this. The assembling the sequencing machine here is actually simpler than using Lego bricks, because in Lego, you still have to push those little things together. Here, we just have to dump the liquids together and it just works. So, and then when this enzyme controls the speed of the DNA, then we observed such current traces. And this is about real time here. You can see there is 1 second trace. It was 50 or so nucleotides that passed through there. And you can see that there are current levels that are pretty distinct. And initially this, when you see this on oscilloscope, looks like noise and like, like a blip of noisy trace. And it's like, we didn't have a clue that was a real thing. But we looked at these traces with more and more detail, and we realized that when we did 20 of those blips during which the current dipped down and had this funny leveling structure, when we look at just the current levels, so we just look at not the duration or anything, but just the amplitude of the current, and we mark the amplitude in this plot here as a value. And we did this for any of these 20 traces that went through there. We saw that it repeated incredibly well. The current always had a level that was at this current amplitude, and it was always roughly at the same position. And so the superposition of 20 of these current traces lined perfectly up. These current levels were very, very repeatable. And at that point, we knew we had something that worked. And then, of course, we counted the number of nucleotides that were in the DNA. And you plot them with these current levels that we found. And the current, there were just about the same number of levels as there were as there were nucleotides. Not always. There were a few places where it didn't do so well. So here were 3 C's in a row. There were a whole bunch of T's in a row, and we didn't see them all. There were obviously little places where there were errors. And it's already clear if you have a homopolymer region, so a little region where you have, in this case, 5 T's in a row, you would expect this, that you don't observe all of them, because the current is the same each time you have a T there. And it's roughly the same. But this was the first functional proof of nanopore sequencing, and published by my student Elizabeth Monroe in 2012. But of course, as you can see, there's more than 4 current levels. So ACGT is just four levels that you would primitively expect. And so there's a little bit of physics involved here. And this physics has to do with Brownian noise, KT noise. So first of all, though, the pore has a finite length. So there's actually not quite 1 nucleotide in the pore if it were a totally static picture, but maybe 1.5 or so, or maybe 2 at a time. But then what's much more important is that there is this thermal motion. There is Brownian motion that stretches this DNA. So the DNA is an entropic spring. And when it gets buffeted by all these water molecules bouncing into it, it stretches or moves the DNA back and forth constantly. And it turns out that it moves it back and forth by up to 4 nucleotides. And so initially that was of course discouraging because we had hoped that we would get 1 current value per nucleotide. It wasn't quite so, but again, you don't give up. We measured all the 4 nucleotide combinations that you can possibly get and measured their current profiles as it goes through the nanopore. And we showed that you indeed could decode this de novo. So here's just an example why this works. So if you have the 4 nucleotide combination ACGT, then with the next step, it has to move to something CGT and then some X, some other nucleotide. And so ACGT, which I guess here works out to be 41 picoampere. The next current that it can move to is either CTGA, CG, or T, one of those 4 nucleotides is next, which are distinct in current. And then I had my group, and here comes the only clinical part. So for those that are squeamish, don't look at it. It's one of my graduate students working on this problem to work out that in the end, you end up at 29 picoamperes. So my group became heavily involved in bioinformatics, but solved the problem. At this point, this technology got very attractive to industry and was left for industry. And industry moved very fast on it, but industry is also very fierce, honestly. There was a fork in the road. And when you see a fork in the road, you might want to take the right end. There were lawsuits, patent fights. It was just very stressful. And so while this thing went off to industry, we thought, what else can we do? And so we came up with this thing called Sprint, which is totally amazing. And so from now on, I will tell you what Sprint actually is. So what we do here is instead of sequencing DNA, which we do here with in the constriction, we now concentrate on the enzyme. So instead of going in with an unknown sequence, we just go in with a known sequence and we see what the enzyme does. And there's a whole bunch of enzymes that walk on DNA. There's polymerases, helicases, and others that walk on RNA, reverse transcriptases, and so on. A lot of enzymes that walk on DNA. And so I will later contrast this to other single molecule techniques, for example, optical tweezers, in which you also use a force. And this thing here generates a force of 20, 30 piconewtons. And you have either an arrested enzyme or so, and measure the position of the DNA relative to the enzyme. So anyway, why would you want to look into how enzymes function? One of the things, especially during COVID, we were reminded that you could make, if you understand these enzymes really, really, really well, you can devise medicines or so, or drugs that interfere with the enzyme and make it not work well. For example, SARS-CoV-2 encodes polymerases and helicase. So whenever that damn virus infects you, it's really sneaky because it makes its own replication mechanism. It makes its own polymerase, makes its own helicase. And so if you can interfere with that helicase or the polymerase, you can interfere with COVID and make it replicate a lot slower. And so, but there's tons of other applications why you want to understand these enzymes really, really well. It's not only for drug screening, but for a host of other uses and understanding of it in the medical field or fundamental biophysics. So how does this really work? So here are again a bunch of these current levels. So again, just the current amplitudes, and a physicist knows that what this actually is. You measured the DNA at these every positions that are one nucleotide away from each other. You measure the current, and so you can put a spline through there, and then you can extrapolate if a current occurs somewhere in between, not at full nucleotide positions, but occurs somewhere in between, then you can argue that the current would be somewhere in between. And that is sort of the principle of this new method of looking at a single molecule study. Here's another demonstration of how it works. So here's the current pattern that you record. And here is a piece of that spline that I just showed you. So whenever you have a current here, you go with the current, go to the right, and look up on that spline function what the position is. You go to the next current level that you apparently see, you go into the spline function and look up what the current is, what the position is. And you go along, and therefore you're practically linearizing this relatively complicated ion current trace, but you keep the kinetics, so the duration of each level into the plot here directly, but convert the current into position. And that's really the essence of how you now get a position versus time curve, just like optical tweezer folks do, or magnetic tweezer people do. You can get these a very, very high precision position versus time curve. Yeah, Will. Yeah, so whenever, for example, you have a low derivative, it's a little bit ambiguous of where you're actually at. So what you want is some DNA that has lots of big derivatives, because then your precision gets very good. Yeah. Exactly. Yeah. So the spline is not ideal, but in reality, it's a low pass kind of filtered curve. We've done lots with modified bases, but in this case here, in this particular picture, that's actually crazily modified base. This is the X here is an A basic, so it's not ACGT. It's just the backbone of DNA. So we had some DNA that had no residues, no side groups, and so that, of course, super exaggerates the currents and makes us particularly sensitive. But yeah, so you can do it with all kinds of DNA, all kinds of sequences, and so on. But what's really amazing here is that the distance between nucleotides, so here on single-stranded DNA is about 600 picometers. And what we saw here in this particular thing is that there were steps that only went by half a nucleotide. And if you look at the noise on these traces here, it's actually very little. So therefore, the positional precision is much, much better than even those 300 picometer steps. This was totally unheard of in the single molecule community, that you could potentially do that. Okay. And so that is the essence of our new single molecule picometer resolution nanopore tweezers. If you don't call it tweezers in biophysics, you're nobody. So we call it tweezers, even though it doesn't look like a tweezer or anything, but you got to call it tweezers. Biologists love tweezers. And so we used it on a whole bunch of enzymes. And so what comes here is like a smorgasbord of enzymes on which we showed off our technology. So this is a helicase. It's a very common helicase. You have held through it in your bodies right now. It does its job, but this particular one that we chose was one that came from a thermophile bacterium that is very tolerant to radiation. And so it's a very, very hardy helicase. We could use it at high salt, which was really useful for us because the ion current is better and better the more salt you have in there. So this helicase worked really well for us. It comes from a super family 2, and what this indicates to you is that these enzymes are grouped in super families, and all these super families that they look similar and they work similarly. So this thing here represents a whole bunch of helicases, therefore. And so here's the position versus time graph that we recorded, and you can clearly see it makes nice steps. You can already see that some steps apparently look like they're longer. If you look carefully, it is that helicase that makes 2 steps per nucleotide. That was entirely unheard of, that you could see this, because in optical tweezer studies of similar enzymes, people had never ever seen that there were sub-steps to every step. So it takes 2 steps. And then we gave this, we looked at the time duration of each step. And we did this as a function of ATP. So ATP is the fuel that these things like. You need to give them the fuel. And so we plotted the duration here as a function of position, and then the 2 curves for 2 concentrations of ATP. As you would expect, if you give it a lot of ATP, it would move faster. So the durations per step will be shorter. And if you give it little ATP, then it moves slower. But what we saw was that this trace of the high ATP looked like it's very jaggedy. And what it turns out to be is that one of those 2 steps is ATP dependent, and the other step is ATP independent. And so you can plot it as a ratio of the ATP level, and you can see that very clearly this step here is ATP independent, but every other step is ATP dependent. This, again, were experiments that people could never do before. Then my student went ahead and made a distribution function of all the durations in the ATP dependent step and in the ATP independent step. And enzyme experts amongst you will know that if there is one sub-process involved, it's just an exponential function. But if there's more than one sub-process involved, then it's not an exponential function, but has a distribution as what we found here. What that meant was that for every half step, we actually saw that it was underneath that yet another step. And so for the chemists amongst you, we worked out the full kinetics of all these steps and all the things that happen chemically during the process. So it was even better than finding, these half steps. We saw that there were sub-steps to those. Then, yeah. So it fits best with 2, but there could possibly be more in there, but they would be so short, those steps, that it would not even show up in here. But there could be if there's microsecond steps, we wouldn't quite see them, but they could be in there. We looked for them, but there was nothing here which you would see near the origin, but we couldn't. Yeah, well, those bins here are fairly narrow, as you can see. Yeah, but it's at some point you won't get more information just because our detection bandwidth will cut things off at really high frequencies. But there could well be something underneath it, but that even escapes us. But mind you, this is a factor of 1,000 better than optical tweezers. So, but my student didn't give up on this either, and he wanted to look at this stupid thing called backstepping. And I went like, John, if you look at backstepping, this is like one of the stupid error modes of this enzyme. It's just not interesting. He was adamant, wanted to look at backstepping, and it turns out this was really good actually to look at backstepping, because if you look at the probability of the backstep, so it's propensity per time to step backwards, it depended on the sequence. So this is color coded here. So whenever there is a whenever there's a C, so here's just C's, right? There's very little backstepping. Whenever there's an A in there is higher backstepping. So for these two here, there's higher backstepping, and that pattern seemed to repeat itself. So we found that the backstepping kinetics is highly sequence specific. That was, of course, quite exciting. So cytosine suppresses backstepping. Adenine and thymine tend to promote backstepping. So here we look at it as a function of the position measured from the constriction of MSPA. And you remember that the constriction is sort of below MSPA by some distance. So the constriction is somewhere over here. And then we saw that the backstepping rate over the size of the DNA or the region of the DNA that was within the enzyme actually had all these fluctuations and had a large variability in backstepping. If it was past the enzyme, the backstepping rates were, of course, could not be seen. Likewise for the duration, so that the time that it spends on one of these levels depended on the A versus C or G or T identity in this case, at position 17. So here's the generic physics approximation of something, right? Enzyme is just approximated by a sphere. But the interact, so and the grayish black stuff here, that's the DNA. And then the colorful things are the amino acids with which the DNA interacts. And there's a whole bunch of places where the DNA interacts through base stacking, for example, with the nucleotides. And so you can then go ahead and correlate the backstepping with particular positions in the DNA associated with certain amino acids in the protein, in the enzyme. You know, I always have my thought on the money here. Our system therefore has up to 2 readers. One of them is the ion current reader, as I pointed out earlier, is the essence of normal nanopore sequencing. But the enzyme also has sequence specificity through its kinetics, through the time structure of the whole thing. And so the idea here is to feed back onto DNA sequencing again by making this second reader, which is a totally independent reader, more sensitive to the sequence. So enhance the differences that you would have in this plot, for example. Okay, and since it was so fun to play with enzymes, we took another enzyme. Here's another. This is superfamily 1 helicase, and we did this together with TJ Ha. So here now you can already see we're starting to collaborate with very famous biophysicists. TJ Ha had already engineered this particular helicase with what's called a processivity clamp. Processivity means this helicase stays on the DNA for a long time. That's called processivity or high processivity. And so it's an ideal enzyme therefore to use because we want it not to fall off and the experiment to end quickly. So we use this PCRAX, which stays on, but we also use it on a very, very long piece of DNA. So we made some sort of measure stick DNA that had particular structures that repeated and 6.4 kilobases long, and it stayed on the enzyme stays on the DNA for long distance. Measure these ion currents, convert all this into position versus time, and since I can't plot it on the screen here at all so that you can see anything, I broke it up into its 64 repeat sections. And you can see that in these repeat sections, there's already places where when the DNA arises at this sequence here, it looks like it stays on there a little bit longer. So again, it hinted towards sequence specificity. So we were able to operate this particular helicase in 2 different modes. And so what these helicases actually do is they're like inchworms. So they move like an inchworm. So this is PCRA here. Since I know gravity, I used gravity as a force here. So what this graph really shows you is you can run this thing in a force opposing mode, where the poor helicase here has to do more work because it's the weight here, the force doesn't help it. In this case, it's force assisting. So the little inchworm moves in this direction, lets the DNA down through the pore, and the force just helps it. That's force assisting. And sometimes we give it also the job that a helicase should really do, is namely separate double-stranded DNA. So there are these 3 modes that you can run it in. I'll come back to this in a second, but what you can see here is that in the force opposing direction, we have the particular parts of this helicase sit on the pore, while in the force assisting direction, the other end sits. So here's the green and here's the blue end, okay? And depending on this, we see different domains moving that have to again, something to do with when and how the ATP is hydrolyzed. So, well, this is probably too much to understand in a small amount of time here. What you could definitely see is that there is a large dependence on the DNA sequence. So here plot the dwell time and note it's a logarithmic scale. So this helicase in some places moves very fast by up to a factor of 10 different than in other places. And also plotted here is the DNA moving in the same direction as in, or the helicase moving in the same direction as it is for unwinding, as in its translocation direction. So the two graphs here, the blue and the red graph, are different by the fact that in one case, it has to also split the DNA apart. When it has to split the DNA apart, of course, it moves slower because there's there's more resistance to it. But what's interesting is that the durations in places in the just translocation mode, when it doesn't have to unwind something, tracks very well with the in positions where it has to in a trace where it has to unwind the DNA. And what people always thought is that in the double-stranded unwinding, what matters is what's in the DNA that has to be unwound. So if there's CG pairs versus AT pairs, but this shows that most of the sequence specificity actually arises from the primary strand on which that helicase is running. Also something that people had never seen before or were unable to look at. Okay. Another thing we did with this is we plotted just the velocity of these pCRA molecules on the DNA, and we plotted it as a function of the force. So in force assisting, its values on the right, and force opposing is on the other end. In force assisting, of course, there are two modes. One of them where there's no double-stranded, and another one where there's double-stranded and it's slower. What you can do here is you can just extrapolate this to the stall force here, also for the purely translocase mode, so where there was no double-stranded DNA involved, and you can work out what the stall forces are, or what the force is, or what the speed would be at zero force. But what was particularly interesting and puzzling or exciting to the biologists is that there's this large spread in velocity, even though it's the same molecule, it's the same amino acid sequence, presumably very similarly folded, but there's this large spread. And what we believe is that this is static disorder. So that's a thing where we don't quite know what happened yet, or what happened to the enzyme. It could be that it's not folded exactly the same way. It could be that it's oxidized a little bit and its functionality is inhibited. In very rare cases, we see it switch its behavior during the experiment. But anyways, this is something that really excites biophysicists or biologists apparently, and may have something to do with cellular aging. So in you always wonder, why do you need to constantly replenish your cells? Now, that might be the reason here, because some of the helicases or polymerases just break and age. Those things break like cars break eventually too. So that is an interesting thing. And so in the absence of plenty of time, I will jump over a few things here. I'm happy to come back later if you like. This is an RNA polymerase where we also see interesting behavior. I'm going to jump over this because I want you to understand finally why the system is so sensitive. So this is, I'm also going to jump over this, but this is this helicase that is in COVID. So when everyone had to shut down their labs, I didn't want to shut down the lab, and I thought, what can we do? And so we did COVID research, and we were allowed to operate during the COVID crisis, and we looked for drugs that can inhibit these enzymes. Turns out that some of the drugs that are around actually are enzyme inhibitors, or the only drugs for viruses are enzyme inhibitors. And so studying these enzymes in our single molecule assay with various drugs, that's really the, you can do drug discovery basically. But let me tell you, before time is totally up, why this method is as sweet as it is. So here's the best thing here. This is optical tweezers. Got Arthur Eshkin a Nobel Prize, I think in 2017 or so. And works extremely well. It's a workhorse. Any self-respecting biophysics department probably has at least one person working on optical tweezers in there. And this is the best trace that optical tweezers can record. And so you can see their time scale is seconds. The one nucleotide steps aren't really all that visible. This is their very best trace. I looked, in their supplement, they have the honest traces. There's many of them, and most of them have hardly any steps in there. So they can just barely see the steps, but they have to do yet one more nasty trick. They have to starve, in this case, this polymerase of its building blocks. So give it a lot less than in vivo. And here's our method. Yeah, so because their bandwidth of the measurement is just so slow, so low. Here's ours, and you can see these lines are just look like stair steps. I remember there's a famous biophysicist that is responsible for this recording here, and at one conference he stood up and said, "Jens, it would be really nice if you would ever show us the noise." And I go, "Well, Steve, you got to put your glasses on because the noise is actually on here." This honestly has a noise in there. You just can't see it because it's so little. If you were to use optical tweezers at the same time interval that we have here for in which everything happened, then this is what the optical tweezer trace that's equivalent to this section here would look like. So no comparison. And so what's the reason for this incredible positional as well as time sensitivity that we have? So here I show you an optical tweezer setup. This is like the best optical tweezer thing that you can do. You trap two polystyrene spheres and two lasers, sits on very sophisticated vibration isolation platforms. It's about a setup that fits on two of these tables, I would say. Probably costs a couple $100,000 worth of lasers and optics and cameras and whatnot. And so I show you this here, and I also show you our setup. You have to look careful for it because it says fly spec here. That is our setup. So our thing is actually so much smaller. and therefore so much more adequate to make measurements at short for these very small systems. So to drive it home a bit more, the reason why the optical tweezer setup is not working as well as our setup is that you have this long tether here, long being 1.5 micrometers. You can't bring these two spheres so close that you could have a much shorter tether, because you cannot scrap two of these polystyrene spheres in, you have to have some optical separation, basically. But the problem is that there's this long tether here. And this is like, if you have a long slinky of some sort, you move it a little bit. By the time you try to measure something that's very small on the other end, it just doesn't work out, because the long slinky kills your bandwidth of the measurement, and also is subject to a lot of KT noise, all the fluctuations in the water that happen here. So this is to drive it home even more. This is the optical bead on which this thing sits. This is the DNA, the long tether, and here's our system. So this tether here is a lot shorter, and it's really the reason why why our method works so much better. Both of these methods put forces on there that are of the same order of magnitude, tens of piconewtons or so, but this method can resolve the position of the DNA just so much better. Here's a log plot of the step size versus the state duration that you can resolve. So if you want to resolve some, if you have one, or if you have 1 millisecond of time here, then you can resolve everything basically on this line. So for example, with our method, you can resolve 1 nanometer within a millisecond easily. And OT stands for optical tweezers, MT stands for magnetic tweezers. And so you can see if you have just a millisecond, then with optical tweezers at the best, you can maybe resolve 10 nanometers. So that really tells you that there are a lot of new enzymes and so on that you can measure with this new technique that are now available for measurement for this assay. So there's yet one other thing that our method gives you for free, and that's also the sequence of the DNA that goes through there, which is why we could discover the sequence dependence, for example, because it's actually at the same time it just sequences. It just is nanopore sequencing. Here's a list that is constantly growing of people that are now switching to our method, and there's a few big names in here that are retooling their labs. So, well, in summary here, I've told you that this is a real-time method with sub-angstrom position, millisecond time information, sequence context in the active site, and so on. We get detailed kinematics and dynamics in conditions that are similar to in vivo. The sequence dependence is found in just about every enzyme, and we're trying to now parse out, does nature actually use that information of sequence dependence in enzymes? We're also trying to figure out if the static order has something to do with cellular aging. Oops, and here are the culprits of that do this wonderful work. And this is what they set up. This is what this truly looks like. This is not an acre, or a large optical table of some sort, but it's a cheesy little setup held together with tape and alligator clips and so on. So that's No, it's not on the floor. It's actually on a piece of copper, which I had left over from my gravity lab. But you know that it's actually easy to do. You don't need to have expensive setups for this. Thank you.